Welcome to Anatomy Made Easy. I am Dr. Amrita Bharti. Today we are going to start with the topics on genetics. The first topic that we take up is cytogenetics. What is cytogenetics? It is a branch of genetics that studies the number and the structure of the chromosome. Following methods that it involves are karyotyping, FISH, buccal smear for fluorescent bodies, study of the sex chromatin on the bar bodies. What is karyotyping? Karyotype. What is a karyotype? First of all, we'll see. It is a micrograph of complete chromosome set of a cell. Karyotype is also known as an ideogram. Karyotyping is a method of obtaining a karyotype of the cell. It is arranged according to the standard classification in a decreasing order of length, and at the end it is followed by the sex chromosomes. For chromosome analysis, the karyotype is prepared from the arrested cell division at metaphase. The best phase to study these chromosomes for karyotyping is the metaphase because it is the most condensed. The chromosomes are most condensed in this phase. What is karyotyping? Karyotyping, we use the method of sample collection. We do the culture and then we incubate it at 37 degrees centigrade for 3 days. We use the Colchikin block to arrest the division in the metaphase. Harvesting of the chromosomes are done. After the slide preparation, banding of the chromosomes are done, and then staining methods are used, followed by the karyotyping and arrangement of the chromosomes in a specific pattern. The various methods of the karyotyping can be remembered as by the mnemonic BP is high in sleep. So, this is a mnemonic used to remember blood collection, planting, incubation, harvesting, and staining methods of karyotyping. First of all, what is done is the sample collection. So, for sample collection, we take around 5 ml of venous blood from the peripheral veins. Then they are collected in the heparinized injection syringes. Then phytohemagglutinin is added to it and the culture medium is added. It is cultured at 37 degree for almost 3 days. Colchicine is added to arrest the cell division at mitosis with hypotonic solution wash. Then cells are fixed and slide preparation is done by mounting it on the slide. The trypsin is added to digest the protein content and proteolysis is done, followed by the staining with the James's stain. And then the chromosome is stained in the metaphase. Further, the karyotyping is done according to the decreasing order of length and the size of the chromosomes, followed by the sex chromosome at the end. Therefore, a micrograph or a ideogram is produced, which is known as the karyotype. Coming to the protocol for karyotyping, sample collection. The samples required can be from blood, it can be from the chorionic villi sampling, it can be from the amniotic fluid by amniocentesis, bone marrow samples, fetal blood, or skin fibroblasts. Sample has to be collected in a heparinized syringe. Heparinized syringe because it is an anticoagulant. It prevents the coagulation of the blood. Then the culture is set up with this method is known as planting. Cells from the collected samples are transferred to culture media. Culture media contains the fetal bovine serum. This provides the nutrition to the cells. Phytohemagglutinin, which is a mitotic agent for inducing the mitosis, antibiotics to prevent the bacterial growth. Incubation is done at 37 degrees centigrade for 72 hours to allow the cells to undergo mitosis. Then harvesting is done by adding colchicine block. Colchicine is added in small amount to block the cell division in metaphase. Harvesting of these cultured cells are done and then it is washed, centrifuged and separated out the pellets of small cells. These cells are put in a hypotonic solution, RBC is removed, and the cells are allowed to swell in the hypotonic solution. Further, the chromosomes are separated as the chromatids. These cells are fixed with a fixative, which is actually a mixture of glacial acetic acid and methanol. This helps in the fixation of these cells. Further, the slide is prepared from the drop of the fixed cells. Trypsin is added to denature the proteins by proteolysis. Staining is done by the use of various stains like the Jamesa stain, and the method that is used for banding is known as G banding. There are various other methods known as the Q banding, R banding, C banding, or the nucleolus organizer region staining. So these are the various methods of staining which is used for the staining of the chromosomes. After these staining, the slide is observed under the microscope and it is microphotographed. The chromosomes are arranged in decreasing order of length with the sex chromosomes at the end. Photographed chromosomes are arranged in the following manner according to the length of the chromosome, position of the centromeres, presence of the satellite body, presence of the specific bands. According to the Denver classification, the chromosomes are arranged into 7 groups, which is numbered from A to G, like A, B, C, D, E, F, and G. While arranging the karyotypes, all the chromosomes are arranged in pairs in their corresponding groups, except the X and the Y chromosomes that are arranged at the end. These are the sex chromosomes, and X is always followed by the Y chromosome. This is a normal male karyotype. The arrangement of the chromosomes can be seen in the decreasing order of the size of the chromosome. So 1, 2, 3, 4, it's a decreasing order. And at the end, you can see there is X and Y chromosome, which actually is the sex chromosome. This is the normal female karyotype, 46XX. Again, the arrangement can be seen in a decreasing order of the size and length of the chromosomes. And at the end, we have the XX chromosome. Denver classification of chromosome, we have A, B, C, D, E, F, and G groups, and the chromosome pairs are arranged in the following orders. They are also arranged in the order of the centromeres present, on which it is classified as metacentric, submetacentric, acrocentric, metacentric chromosomes. Use of karyotyping is done in clinical diagnosis of cases with the chromosomal aberrations, clinical confirmation of cancer cases showing various translocations, for example, detection of Philadelphia chromosome in chronic myeloid leukemia, prenatal diagnosis for chromosomal aberrations, diagnosis of chromosomal defects in infertility cases or in recurrent abortion, gene mapping, and various cancer cases. For detection of single nucleotide change in DNA, polymerase chain reaction is used rather than cytogenetics. Coming to banding. Banding is a staining technique for chromosomes. Stains chromosomes along the longitudinal axis. The chromosomes are stained in dark and light bands. Band is a part of chromosome which is actually clearly distinguishable from its adjacent segment by lighter or darker with various banding methods. Why banding pattern is studied? It allows to see the smaller pieces of the chromosome so that one could identify smaller structural chromosome abnormalities not visible on a routine analysis. Classification of various banding techniques is based on the guanine, cytosine, and adenine and thymine rich regions and also constitutive heterochromatin regions. Always metaphase chromosomes whose size has condensed and whose diameter is increased are used for the chromosome banding studies after fixing the stage. These are the various banding techniques: Q banding, G banding, N banding, and C banding. Q banding is the oldest technique. Scientists associated with this is Kasperson et al. James staining is the most commonly used method by Summer et al. The N banding is by the Matsui and Sasaki. The C banding is the latest banding technique by Linde and the Lawson. G banding is also known as Jamesa banding. It is the most commonly employed method for karyotyping, and the need for the banding method is to differentiate between the chromosomes of the same group, for example, chromosome 4 and 5, and also to identify the various chromosomal anomalies like addition, deletion, inversion, and translocation on the chromosomes. G banding can be done on slides of chromosomes after treatment for 10 to 20 seconds with trypsin, which will partially digest the histone proteins by proteolysis. Then Jamesa stain is added. Densely stained region of chromosome is known as a dark band. It is rich in adenine and thymine. Lightly stained regions of chromosome are known as the light bands, which is rich in guanine and cytosine. G banding techniques, we have the chromosome. Addition of trypsin or the protease is done to denature the proteins. It is treated with GMSA and the banding is done. Interaction of DNA with thylacine and eosin components of stain brightens the sulfur-rich regions, forming methylene azure, methylene violet, methylene blue, and the eosin. R banding is known as reverse banding. Here it is reverse of the GMSA banding. The slides are preheated. The proteins can be denatured by heat or hot solutions or hot salts. GMSA stain is added. The bands are reversed as in GMSA banding. So the band that was dark band here becomes the light band, and the light band becomes the dark band. So we have a dark band which is rich in guanine and cytosine, and we have a light band which is rich in adenine and thymine. The other method for chromosome banding is Q banding. The chromosomes are stained with fluorescent dye known as quinagrine mustard or quinagrine dihydrochloride dye. It is useful for studying heterochromatin of chromosomes 1, 9, and 16 and various polymorphism and human chromosome Y. The same results can be obtained as in G banding. Here we have dark band with adenine and thymine rich and light band which is guanine and cytosine rich. C banding is the method in which the DNA of the chromosome is denatured with barium hydroxide, acid, alkali, or heat. It is then stained with the GMSA stain, and here only dark bands are obtained, which is useful for studying constitutive heterochromatin. It is used for staining human centromeric chromosomes. Use of banding techniques can be for the karyotyping of an organism, for size, shape, and number of chromosomes in a cell, for determining the types of the cells, whether it is types of the chromosomes, whether it is acrocentric, telocentric, or metacentric chromosomes, and for determining various chromosomal aberrations. What is FISH technique? FISH or fluorescent in situ hybridization assay. It is a cytogenic technique that utilizes the property of DNA probe to bind with its complementary target DNA sequence. The method for visualizing specific locations on a chromosome by a fluorescent complementary probe. Basically, it is aiming to detect the chromosomal abnormalities. DNA probes are tagged with fluorescent substances, which may be a radioactive substance. In this, the cells are collected from the tissue. Fixation of the cell is done in the formaldehyde, causing either protein-protein or protein-nucleic acid crosslinks. Design of RNA or DNA probes. DNAs, including random cuts and nicks, are used. Endonuclease enzyme labeling the probes provides special nucleotide with fluorescent proteins, ligase, seals the neck. Denaturation of the probe is done in cells of interest. It is done by heating the cell at 95 degrees. Temperature is cooled down, allowing the labeled probe to specifically bind to the DNA sequences. Then the hybridization or the cooling down method. Once the hybridization has taken place, all probes that are not bound to any DNA are washed away. Fluorescence microscopy results can be analyzed under the microscope. If the probe is designed so that it can only bind if it is 100% complementary to the gene, we expect to see an extant loci under the microscope. But if any mutation has occurred in this gene, no hybridization is possible. This is possible in interphase fish. In metaphase fish, again, the culture is done with the colchicine block. Harvesting of the chromosomes are done. Metaphase preparation is done of the chromosomes. Hybridization with the DNA probe is made and the observation is done under the fluorescent microscope. So with this we have finished lecture on cytogenetics. Next we'll be taking up lectures on chromosomes and types of chromosomes. Thank you.